Let's go ahead and finish off this chapter. We were just talking about immunoglobulins, which are the technical term for antibodies. And one of the things that's important is this constant region of our immunoglobulin is what gives us our various antibody classes. So let's take a look at the 5 antibody classes we have. Immunoglobulin D, which is what Ig stands for, M, G, A, and E. Immunoglobulin D is a monomer, so we have this lovely Y here. It is very important in activating our B cells and is actually our B cell receptors. So on our naive B cell, the little Ys that we had on the surface were actually immunoglobulin D. IgM is a pentamer. It is the first class that is released during our primary immune response. We'll get a little bit during a secondary immune response, but nothing like our primary immune response. So as a pentamer, it is 5 Ys and has a high number of binding sites. And it takes a long time to actually construct this. This is another reason why our primary immune response is so slow, because we first start making IgM before we make IgG. And IgG is the star of the story. It is a monomer and the main antibody of our primary and secondary immune responses. So what's really nice about this guy, he has the ability to cross the placenta and therefore protect unborn children, and is also the immunoglobulin that is used for our passive immunity when we actually inject somebody with anti-venom, for example. IgA is a dimer. So we have our Ys here. And it is protecting our mucosal surfaces. So we're going to find it in our lacrimal secretions, in our saliva, along the gastrointestinal tract. That is the immunoglobulin that is associated with our mucosal surfaces. IgE, that's a really sad one. It's too bad that we actually produced this guy. Again, it's a monomer, and it unfortunately will bind to mast cells and basophils, and whenever we get activation of mast cells and basophils, it's because we have unfortunate binding of the allergen to this thing, and we'll have degranulation with the release of histamine and, of course, some heparin whenever that occurs. So how do these antibodies from our plasma cells actually work? Well, they have the ability to actually create a kind of a targeting action on cells. And so they will mark the cells for other organisms to attack or even complement to attack. So target is kind of like marking cells or pathogens, and let's take a look at some of this activity that we actually see here. Neutralization isn't a targeting thing. Neutralization is our simplest mechanism. It is where the active sites will attach to the toxic sites that we'll see with our toxins or the really bad sites on viruses. So if it attaches to the really nasty parts of the virus or toxin, in essence, we are blocking those active sites, and that is creating an inactivation of the really nasty nature of that pathogen. Indeed, neutralization is what happens when we inject antivenom into an individual. It will block the active sites. There will be another aspect that we'll talk about in just a moment. Well, actually, let's go ahead and go to it. Once we inactivate rattlesnake venom with our antivenom, we will then have this precipitation actually occurring, where things were more in a soluble nature, but because we've now inactivated it, we've got a whole bunch of immunoglobulin attached to this molecule. It will cause the soluble molecule to become insoluble, and settle out, and will make it easier for our macrophages to actually go to and gobble up and get rid of. Agglutination, this is indeed also possible because of the bivalent nature of the antibodies. And what we have here is we have cross-linking. Of the various antigens that this antibody is responsible for identifying, and this will cause formation of large complexes that settle out and are therefore accessible. He's accessible. I think, oh, excuse me, my spelling is real off on this one, so forgive me on that. But they are accessible for our phagocytes also. However, they can become so incredibly large that they actually block up blood vessels. And this is what happens with blood transfusion reactions. That's why we call surface markers of our red blood cells agglutinogens, because of the agglutination process. So let's say we are type A, so we have the A agglutinogens on the surface of our red blood cells, and due to the bivalent nature of our Y's here, or of our immunoglobulins, it's pretty sad looking. I don't think I can make it better though. Nope. We can then cross-link and we can make these really large complexes that are very heavy and precipitate out, and that's what gives you the actual visible agglutination reactions that you see if you're just doing some very basic blood typing in a lab situation. Complement fixation, we've already touched on that. That is our main defense against bacteria and incompatible blood. You go, well, wait a minute, you just talked about agglutination with these, with blood that we should not be encountering because we have antibodies against that blood type, which means it's obviously not our blood type. But complement fixation is a tagging process. So it tags cells for destruction. And remember, it does this with MAC, membrane attack complex. And that's an important thing when we actually talk about complement fixation. So let me just take a little bacterium here that has these various surface markers on it. I'm just going to make little dots for surface markers right now. And we have antibody that's that recognizes this, and so it's tagging it. And that is important because we have a whole bunch of antibodies attached to the surface of the bacterium that now make it more easy, or it's easier for MAC to recognize it, and then the complement proteins that make up MAC then can attach to the surface of our bacterium and cause polymerization in a big hole. Well, take a look at what I just drew up top with the agglutination. We're having a whole bunch of antibodies attaching to our cells here, and this too will cause the very same process where MAC, which was floating around, now sees a bunch of antibody tagged cells that are clumping out, or even before they clump, and then you get the MAC attack here on this cell and makes a big hole in a red blood cell, and then we have hemolysis. We have hemolysis where the contents of the red blood cell is then extruded out into the surrounding environment. All of this is very bad in blood transfusion reactions. So let's take a look here. Binding of antigen. Binding sites on an antibody with the antigen will cause neutralization, which is our toxin thing that we just discussed. Agglutination, because of cross-linkage, because of the bivalent nature of our monomers. And then, of course, precipitation, where we actually have much smaller molecules complexing with our antigens. And so we do get a smaller type of material that actually settles out when you compare it to agglutination. Agglutination is big stuff settling out, precipitation, small stuff settling out, both due to the bivalent nature of the antibodies and cross-linkage. Complement fixation. So we actually can have the complement will be activated, which is what it's doing right here, because of the antigen attaching here. I'm not doing a very good job, but this will then bring forth MAC to attach to the cell. And opsonization, remember that we actually can have complement attached to the bacterium to make it more attractive for phagocytes. And activation of natural killer cells for apoptosis, excuse me, and we won't hold you actually responsible for that one. Homeostatic imbalances, so problems with our immune system. You can have immunodeficiencies as our first topic, or we can have hypersensitivities, and that will be our next topic. Immunodeficiencies, they can be congenital, which means you were born with this, so there's something missing for your immune system to function properly, or you can acquire, after you're born, something happens to you that makes your system not respond in the proper fashion whenever exposure to an antigen or pathogen occurs. Of the two immunodeficiencies, I would like you to know about severe combined immunodeficiency disease. SCID is the abbreviation. And this means that we have an absolute deficit of T and B cells. So no T cells, no B cells, and so that means that you are not going to survive unless you're the boy in the bubble. So you have to live in a sterile environment where you have no encounter of any pathogens in order to survive. Because if you have no T cells, you have no B cells, then that means that only your innate mechanisms are protecting you. And so you cannot be introduced to pathogens which could possibly breach your primary defense, which is our surface barriers, and then secondary defense, which is our cellular and chemical methods of protection. Acquired immune deficiency syndrome, or AIDS, is a really bad actor because this virus loves T helper cells. So HIV virus attracted to and attaches to T helper cells, then invades and replicates the virus and destroys the cell. So viruses can then be sent out to find another T helper cell to invade and ultimately destroy. And so the HIV virus loves T helper cells and preferentially infects them. If you infect a T helper cell, which is responsible for co-stimulation of all of our lymphocytes, whether they're T cells or B cells, you have effectively inactivated or shut down both arms of our immune system. And so you have definitely some immunity because remember we have all these memory cells out there that they have to go find and destroy that are T helper cells. Hypersensitivity is also known as allergies, an abnormally strong immune response to something that's not really a threat. There are two main types, immediate and delayed hypersensitivities, but we are only going to talk about the immediate or the acute hypersensitivity. And this is an allergic reaction that begins almost immediately after contact with the allergen. What's important to realize, in order to have this immediate hypersensitivity, you actually have to go through a primary and secondary immune response. So this initial exposure to the antigen causes a primary immune response. And for some darn unfortunate reason, the antibody that is produced in this initial exposure to the allergen causing this primary immune response and causing this antibody production is IgE. And so our plasma cells will be producing IgE antibody, which then goes out and actually attaches to the surface of our basophils and mast cells. And that is called sensitization. So when you find a basophil or a mast cell that has IgE surface antibodies all over it, it has then been sensitized to whatever the allergen or antigen was. Then, unfortunately, when you get re-exposure to this same allergen, then it will actually land on the receptor portion of our IgE, and it will cause the mast cell or the basophil to undergo exocytosis of their granules, or degranulate, and release histamine and heparin and cause this unfortunate cascade of inflammatory effects. I wanted to talk about the word anaphylaxis. An is against, phylaxis, protection. Prophylaxis is for protection, and anaphylaxis is against protection. And this anaphylaxis is due to the allergen cross-linking with IgE on the surface of our mast cells. And basophils, but basophils are in the bloodstream. And the first thing that we'll encounter is typically soft tissue unless we get a needle into the bloodstream. And so the first cells that are typically going to cause degranulation is basophils. And floods soft tissues with histamine and heparin. Histamine, indeed a potent vasodilator, and that is going to increase the leakiness, and so we'll get a lot of fluid invading the soft tissues. And other inflammatory chemicals will also come about because of this. And give you hives, anything that will give you redness and swelling, swelling of the airways, blocking them off, asthma, swelling of soft tissue beyond just hives, where you actually eyes, nose, throat, mouth, and lips all swell up and close off even your airways. Another problem with our immune system or homeostatic imbalance would be autoimmune diseases. And autoimmune is a problem when we lose recognition of self. two main ways this happens is a virus can change the cells on their surface, or we can have an antigen of a pathogen that looks a lot like us. So let's first talk about how a virus can actually change a cell. So a virus invades the cell and alters the surface self-antigens, and it invades the cell and gets placed into the MHC1 flagpole, so to speak, and that now indicates that the self cell is no longer the self cell. This is what happens with multiple sclerosis and insulin-dependent diabetes. What happens is the virus is specific for the cells that, when they are destroyed, causes multiple sclerosis. And remember, the virus, that the HIV virus likes T helper cells, recognizes them, and preferentially infects them. Well, the virus that causes multiple sclerosis preferentially infects oligodendrocytes and destroys them, giving us the multiple sclerosis. And the virus that causes diabetes does so because it likes and preferentially infects our beta cells, and therefore our immune system will recognize these infected cells and take them out. And that is a problem because we need these cells to prevent diabetes or to keep us from having multiple sclerosis. Antigens similar to self, that's confusing our immune system cells, and so we can say this is cross-reactivity, but this is what happens when people get rheumatic fever. Rheumatic fever comes about because somebody has a strep throat infection. With a streptococcus that has an antibody, or excuse me, has a surface antigen that looks just like some surface markers of your heart valves. Also of your synovial fluid, or synovial membrane, excuse me. Or kidney structures. And so this strep throat infection causes you to mount an immune system response to the streptococcus. However, this response is going to include recognizing now a chemical structure from that strep as foreign, and that unfortunately resembles a chemical structure of the molecules of your heart valves or your synovial membrane of your joints, and then your various kidney structures called glomeruli that we'll learn about in a little bit. So finally, we're getting to blood typing and ABO blood groups and Rh factor. ABO blood groups are basically dealing with surface markers on our red blood cells, or remember we call them agglutinogens, and there are more than just the ABO that we know. There are at least 30 common agglutinogens, and the name is what we actually refer to as our red blood cell surface antigen. So RBC surface antigens are also referred to as agglutinogens. We do blood typing to prevent transfusion reactions. And so we'll check for the ABO blood groups and these other agglutinogens, as well as Rh factor, which is another agglutinogen. So we know red blood cells contain, for the ABO blood groups, either A agglutinogen or B agglutinogens. So, or both or neither. So red blood cells, if you're type A, your red blood cells have A agglutinogens on their surface. If you're type B, your red blood cells have B agglutinogens on their surface. If you're AB, you have both A and B agglutinogens on your surface. Or if you're type O, you don't have either A or B agglutinogens on your surface. What's interesting about the ABO blood groups and these surface markers is that we actually have built-in antibodies or preformed antibodies to the blood group that we are not. So we will, if we are type A, in our serum floating around is antibodies to type B. If we are type B, we will have floating around in our plasma, excuse me, I should have said plasma instead of serum. We have plasma that if we're type B, we have antibodies to type A. If we are type O, we have antibodies floating around in our bloodstream to type A or B blood. And if we are AB, we don't have antibodies to A or B because that would fight our own cells. So A is antigen, B is antigen, and anti-serum is referring to the antibody in the bloodstream. So if we are type O and are given type A blood, we immediately have a transfusion reaction. And so what that means, if we're type O and we are given type A blood, we have antibodies floating around in our bloodstream to A and B. And as soon as we are given type A blood, we will have those antibodies all attached to the surface of that blood cell, just like up here, and cause agglutination. So it will settle out, and it's a large, complicated, big clump settling out in your bloodstream, blocking up your capillaries and even your arterioles and venules. In addition to that, because we have all these antibodies attached to these red blood cells, even before they become cross-linked and create these agglutination clumps, we'll start to have complement fixation and hemolysis. So that's what a transfusion reaction is. So because we have preformed antibodies, it's an immediate problem. So that's why type O person only wants type O blood. Type A person only wants type A blood, but can be given type O. And how can that happen? If you are type A and you're given type O blood, that means in the plasma of that blood are antibodies against A. Well, the reason we can do this is because we wash the blood. What that means is that we are washing out the preformed antibodies from that blood. We are just basically working on isolating out the red blood cells and suspending them in a solution that is not the plasma from that individual. So we can give an A person type O blood because there are no surface markers to react to on that blood, and we are not giving them the antibodies that came with that type O person's blood. Rh factor, on the other hand, is a different story. We had 30 for the ABO agglutinogens. Well, for the Rh factor agglutinogens, we have even more. We have about 50. And the most common ones out there are C, D, and E. So those are the ones that are most commonly found. In this situation, antibody is not preformed. So it's not preformed, and therefore we will actually have a typical primary secondary immune response. So what this is important in is that we have a certain set of the population that is Rh positive and a certain set of the population that is Rh negative. And 85% of the population is Rh positive. What that means is that in addition to the ABO surface markers, the cell will either have an Rh marker or not. If it has the Rh marker, it's called Rh positive. If it doesn't have the Rh marker, it's called Rh negative. And that brings us now to the topic about hemolytic disease of a newborn. And remember, mother's antibody crosses the placenta. And that is what brings about the actual hemolytic, which means blood cells are popping all over the place, disease of a newborn. So mother's antibodies or immunoglobulins can cross the placenta. And when they cross the placenta, if there is some incompatibility that mother has been activated for or had a primary immune response, we'll have some problems. But first, we actually have to have a primary immune response. So hemolytic disease of the newborn can only occur, occurs only if mom is Rh negative. If mom is Rh positive, it doesn't matter whether she gets exposure to Rh positive blood or Rh negative blood, because Rh negative blood is kind of like O blood. No surface marker, nothing to respond to. So if mom is Rh negative and she for some reason gets to actually have an encounter with Rh positive blood, she will develop a primary immune response, because this is a primary secondary process. So nothing there, no exposure, nothing happens. But if she comes across Rh positive blood, that will create a problem. So let's take a look at our diagram here. First, pregnancy, antigen D. So that's one of those common Rh factor. Antigen D is introduced into mother's blood because we have our first baby being Rh positive. So mom here is Rh negative. And the time when we actually can accidentally get fetal blood cells to cross the placental barrier, because the fetus is an intact individual and their blood does not mingle with mother's blood. The placenta is that immune system organism that actually causes amnesia. Doesn't allow mom to recognize fetus as actually foreign, and the fetus is actually foreign itself, but the placenta causes immune system amnesia. Well, remember, mom's antibodies can cross the placenta. Ordinarily, fetal antibodies or antigens do not cross the placenta. If they were fetal antibodies, they would be considered foreign, but fetal antigens or abnormal cells don't cross the placenta. The time that mother will actually have her system exposed to stuff from the babies is typically during birth. And so when you give birth to the baby, there's a lot of blood all over the place. It isn't just from the placenta. It is actually from a lot of wounds that the mother undergoes from giving birth. And when we talk about childbirth at the end of the semester, you'll understand a little bit more about this. But when the placenta detaches, it leaves a gaping raw wound inside the uterus of the mother. And also on the way out, there can be a lot of tears that actually occur due to traumatizing areas that have never been stretched as much as they do. And when you have a gaping wound, anything can get into it, including blood that comes from the placenta, which is in and of itself actually fetal blood. That's why they call it fetal cord blood. So placental blood, which ordinarily wasn't crossing and getting into mom's system, suddenly is now crossing, getting into mom's system, and that is fetal blood. And that will cause a primary immune response. Now, that means we are going to develop antibodies to Rh factor. This is anti-D antibodies, but antibodies to Rh factor. Then in the second pregnancy, what happens is mom has antibodies to Rh factor already in her system. And these antibodies can cross the placenta and start to attach to fetal red blood cells because they are Rh factor positive. So if the baby is always Rh negative, this never happens. This only happens if the baby is Rh positive and the mom is Rh negative. And so antibodies are crossing over, they're attaching to fetal red blood cells, and they're causing complement to then attack the fetal red blood cells. They're causing agglutination and complement fixation and MAC attack complex. What we do to stop this process from happening is we actually apply passive immunity to the mother. So she gets a RhoGAM shot. Typically, Rh negative mothers are given a RhoGAM shot at around 28 weeks because then we actually have antibody floating around in mom's system that is actually not the anti-RhoGAM, not the type that will actually cross the placenta. So we can make it a size that will not cross the placenta. But anything that from the placenta that might get into mom's system, it can immediately neutralize. And so that's what RhoGAM shots do. RhoGAM shots neutralize the any sort of antigen that may cross the placental barrier. So RhoGAM. We want a neutralization reaction. We want it to attach to the things that would be recognized as foreign, cover them completely up, so mom will not mount an immune response. This is typically done at 28 weeks. We also then test the baby after it is born, and if the baby is Rh positive, mom's Rh negative, we'll give her another RhoGAM shot. However, this RhoGAM shot will be more of the immunoglobulin G class, which will immediately hunt out and find and immediately also, again, neutralize the red blood cells that could be floating around in mom's system that she would mount an immune response to. And so that's what RhoGAM does. It creates a neutralization reaction for mom. So in essence, mom's system never sees the foreign material to create an immune response. So that's the erythroblastosis fetalis, which is also more easily said as hemolytic disease of the newborn. That concludes our chapter. Please be sure at the beginning of lecture this next week, if you have any questions about this material, do ask them at the beginning, and we will then proceed into the respiratory system after that. Have a good weekend.